Hi, I'm Dr. Hannah Lindsay. I'm a faculty level research associate in the Traumatic Brain Injury and Concussion Center at the University of Utah. My background largely includes training in neuropsychology and cognitive neuroscience, and my research almost always involves some aspect of neuroimaging, but my specific expertise is in diffusion MRI post-processing and analysis. This is part two of a three-part diffusion MRI lecture series for ENIGMA-U and should follow Dr. Emily Dennis's presentation on diffusion MRI. In this lecture, I'm first going to review some general neuroanatomical concepts and then describe the anatomy and function of some major white matter tracts. I will then briefly discuss the diffusion tensor model. Some of what I cover will overlap with information that Dr. Dennis presented in the previous lecture, but I'll be focusing more on mathematical aspects of the tensor model and what its limitations are so that you can better understand the advancements in diffusion imaging analysis that I cover in the lecture that follows this one. The nomenclature used to define a white matter tract is often based on its location and trajectory in the brain, so it's important to understand how to navigate the brain. We refer to different areas in the brain in more or less relative terms, where any region toward the front is anterior to whatever regions are behind it, and anything toward the back is more posterior than anything that's in front of it. The same idea applies to the relative positions along the Y and Z axes as well. So anything toward the top of the brain is superior and anything toward the bottom is more inferior. And finally, anything closer to the midline of the brain is considered to be more medial to anything that is closer to the outside of the brain, which is considered more lateral. When working with diffusion MRI or dMRI data, we are most often looking at slices on a diffusion-weighted image. So you need to know how to read these. Since the brain is 3D, we view and image the brain in 3 dimensions or in 3 planes: sagittal, coronal, and axial. The sagittal plane divides the brain into left and right halves. A mid-sagittal slice is taken exactly in the middle, splitting the brain into equal left and right sides or equal hemispheres. And that's like what you see here in this top row. A parasagittal slice is a slice that is slightly off center or slightly more lateral in either hemisphere to the mid-sagittal slice. The coronal plane divides the brain into anterior and posterior sections, and it's kind of like slicing through a loaf of bread. Typically, when you look at a coronal MRI slice, it's as if you are looking at the face of someone who's standing right in front of you. So in that case, the left side of their face is on your right, and the right side of their face is on your left. So the same is true when we're looking at the slice. The left is on the right side of the screen, and the right side of the brain is on the left side of the screen. The axial plane is sometimes called the transverse or horizontal plane, and it divides the brain into superior and inferior sections. Just like with the coronal plane, left and right are also reversed on the axial MR slices. So it's kind of as if the person were laying on a table in front of you, you're standing at their feet, and you're kind of looking at their brain from the bottom up. And just to be clear, the left and right sides are typically reversed when looking at the coronal and axial slices of the brain, but not when looking at a 3D rendering of the brain. And we'll see quite a few 3D renderings later on. I know that's all confusing, but unfortunately, that's just the way that we do it. Okay, so in this first column here, this is what the brain looks like when imaged using structural T1 weighted MRI. In comparison, in these columns, we see what a diffusion-weighted image looks like also in these same 3 planes and on the same slice as the structural T1. On the black and white FA map, or fractional anisotropy map, brightness indicates the degree of anisotropic diffusion, where brighter areas have greater anisotropy, reflecting the presence of white matter. That makes these really helpful because they only light up on the white matter. So any gray matter regions, like in the basal ganglia here and here, or in the ventricles here, those areas are dark because they consist of gray matter or CSF and not white matter. To help with differentiating between different white matter pathways, we often use a color-coded FA map, where tensor information is directionally coded according to this little illustration there. And what that allows is for any tracks with anterior to posterior trajectories, those are shown in green. Anything moving superior to inferior is shown in blue, and anything running from right to left are in red. Trajectories that occur anywhere in between anterior to posterior, up and down, or right and left are going to fall on this spectrum and kind of be in between. So something that's kind of like superior, but also to the right is going to be pink, for example. White matter tracts in the brain can be categorized into three main types based on the location of the regions that they connect. Commissural tracts connect the left and right hemispheres of the brain. These are shown in red here in this illustration, and as you can see, they start on one side of the brain, cross the midline, and then terminate on the other side. Commissural tracts enable communication between the two hemispheres and play a really important role in functions that require bilateral integration, like coordinating the movements of your body or your perceptual awareness. Association tracts connect different cortical regions within the same hemisphere. And this image, when it finishes spinning around, you'll see the association tracts are in green, and they're only shown on the left hemisphere here. And that's just to indicate that they only connect regions of the same hemisphere. Short association tracts, or U fibers, connect different regions within the same lobe of the brain. And long association tracts connect two or more lobes within the same hemisphere. All of it's within the same hemisphere, but the long ones just span across the lobes. Association tracts are essential for intra-hemispheric communication or within hemispheric communication and support major cognitive functions. And they link cortical areas that are involved in similar or complementary functions. Projection tracts connect the cortex to subcortical areas like the thalamus or brainstem or cerebellum, for example. And they're broadly involved in functions related to like gross motor control and somatosensory processing and autonomic functions. In this image, just like the association tracts, the projection tracts are shown only in the left hemisphere, and they are in blue. The most prominent commissural tract is the corpus callosum, which allows information sharing and integration across the two sides of the brain. And it can be broken down into four parts. The gnu is the most anterior portion, and it primarily connects the two frontal lobes, facilitating communication and integration of higher order cognitive and other executive functions. The body of the corpus callosum is the central portion, and it connects the two parietal lobes. It's important for sensory motor coordination and integrating spatial and perceptual information. The splenium is the posterior portion and connects the left and right parietal and occipital lobes. It plays a key role in the integration of visual and auditory information and supports functions like visual spatial awareness. The tapetum, which you can see in purple, is a smaller component that is sometimes considered part of the body or splenium. It extends laterally from the body and splenium, wrapping around the lateral ventricles and projecting anteriorly into the temporal lobes. Its function is less well understood than the other regions, but it does support interhemispheric communication between the two temporal lobes, and it probably plays a role in language and memory. The anterior commissure is a smaller commissural tract that's located underneath the corpus callosum, and it links frontal, temporal, and occipital regions. It facilitates olfactory integration, emotional regulation, and memory formation by connecting the olfactory bulbs, the amygdala, and medial temporal lobes. And it also aids in visual integration with its occipital terminations. Even though it's smaller than the corpus callosum, it still plays a vital role in sensory, emotional, and visual communication between the hemispheres. And it is particularly important in cases where the corpus callosum may have been damaged. Okay, so these are some of the long association tracts in the brain. We're only going to talk about the long ones. The cingulum bundle runs within the cingulate gyrus, forming a large C-shaped ring from the orbital frontal cortex around the corpus callosum and back down toward the temporal pole. The cingulum is part of the limbic system, and it is involved in attention, memory, and emotions. The frontal aslant tract in purple Projects from the anterior supplementary and pre-supplementary motor areas of the superior frontal gyrus to the inferior frontal gyrus and the inferior region of the precentral gyrus. It is involved in motor, speech, language, and executive functions. The arcuate fasciculus is a lateral association bundle that connects the parasylvian cortex of the frontal, parietal, and temporal lobes, specifically connecting Broca's and Wernicke's areas. The left and right arcuates are very asymmetrical. The left is much larger and it plays a really important role in language, whereas the smaller right arcuate is primarily involved in visual spatial processing, although it does have a relatively more minor role in some aspects of language. The superior longitudinal fasciculus, or SLF, in general connects the dorsolateral frontal and parietal cortices, and like the arcuate, the SLF is very asymmetric. So connections between the supramarginal and angular gyri with the dorsal precentral and middle frontal gyri are only seen in the left hemisphere, and then projections from the supramarginal gyrus to the inferior frontal gyrus are usually only seen in the right hemisphere. The SLF supports efficient processing of visual, spatial, and audiospatial information, attention, awareness, and working memory. The middle longitudinal fasciculus, which is kind of hard to see in these images, but it's right back here in like a light green. It originates in the superior temporal gyrus and projects to the superior parietal lobe and parietal occipital region through the transverse temporal gyrus, also known as Heschel's gyrus, and to the posterior occipital lobe through the angular gyrus. It has only recently really been studied in the literature, and it's therefore somewhat unknown what its functional role is, but given its anatomy, it likely plays a role in higher order auditory processes like the localization of sound in space or the integration of auditory and visual information. The inferior longitudinal fasciculus, which is in blue, light blue, it's the most inferior association tract, and it has fibers that originate from the medial and lateral anterior temporal areas and terminating throughout the occipital lobe. It's involved in facial recognition, visual perception, reading, visual memory, and other visual and language-related functions. The inferior fronto-occipital fasciculus, which you can see best here in dark green, is a very long association tract that connects the frontal cortex all the way to the occipital lobe through the temporal stem, which is this little area right here. It's largely involved in reading, attention, and very efficient visual information processing. Finally, the uncinate fasciculus connects the anterior temporal lobe with the anterior temporal lobe with the orbital frontal cortex, and it is considered part of the limbic system, but its functions are relatively poorly understood. However, it is believed to be broadly involved in emotion processing, memory, and language. Here are some of the major projection tracts in the brain. There are others, including several components of the internal capsule that are not shown here. The first major one is the cortical spinal tract. It's a descending projection tract that connects the primary motor cortex to the midbrain and spinal cord through the internal capsule. It's the main pathway responsible for motor function, particularly that which is related to the face and hands, feet, and lower limbs. The medial lemniscus is kind of the sensory counterpart to the cortical spinal tract. It transmits information about touch, vibration, and proprioception from the body to the brain. It originates in the medulla oblongata and ascends up through the midbrain into the thalamus, and from there up to the primary somatosensory cortex. It's essential for fine tactile perception, vibration sense, and awareness of body position. It enables precise and coordinated sensory experiences. The thalamic radiation, which is this big red thing, it projects from various thalamic nuclei to the cortex and can be separated into anterior, superior, posterior, and inferior pathways, which project to the various lobes of the brain through the internal capsule. The thalamic radiations are involved in the control of cortical arousal and consciousness and play a role in movement disorders, like as in Parkinson's disease. Now, the optic radiation, which is right here in yellow, projects from the lateral geniculate of the thalamus to the primary visual cortex in the occipital lobe. So it is technically part of the thalamic radiations, but it's often studied independently because of its unique organization and key role in visual perception. So it's organized in such a way that the fibers carrying information from the superior or top part of the visual field of the retina travel along the inferior aspect of the radiation, and information from the lower half of the visual field travels along the superior aspect of the radiation. After leaving the thalamus, the fibers fan out as they travel along their trajectory, where the inferior fibers with the upper visual field information move first anteriorly into the temporal lobe before, you can kind of see that here, before moving back towards the occipital cortex. And that makes up what we call Meyer's loop. The more superior fibers with the lower visual field information, those project directly to the cuneus from the or through the parietal lobe. The optic radiation is essential for processing visual input and critical for spatial awareness and details regarding vision. And due to its specialized organization, damage to different parts of it will result in specific visual field deficits. Finally, the fornix, which is this purple guy here, it originates from the hippocampus down here and then arches up, around, and backward beneath the corpus callosum, and then terminates in the mammillary bodies of the hypothalamus. So it starts down here in the hippocampus and then goes all the way around to the hypothalamus. The fornix is part of the limbic system and plays a crucial role in memory consolidation and emotional regulation. Okay, so before I talk about diffusion tensor imaging, there's one more thing related to biology that I think is important to talk about. In order to fully understand how to interpret diffusion data, it is necessary to understand the ways in which water molecule movement is influenced by the microstructure of different biological tissue compartments. And those are represented in a single voxel in this illustration, where we have our extracellular space compartment, our intracellular space, or these cell compartments, and then the axonal fiber compartments. So some of this may be a review, but I'm going to go through it anyway. Isotropic diffusion refers to water molecules moving freely in all directions. And this is typically seen in areas without physical barriers, like in the cerebral spinal fluid. However, in the brain, true free diffusion is pretty rare because of such a dense, complex cellular environment. So we do differentiate between free isotropic diffusion and restricted isotropic diffusion. Restricted diffusion occurs when water movement is constrained by something. So this can happen intracellularly, where diffusion is limited by the cell membranes, or extracellularly, where it's blocked by moving through the cells themselves. So things like cell swelling or immune cell infiltration can hinder the water's movement in the extracellular environment. In biological tissues like the brain, restricted diffusion is often a combination of intracellular and extracellular effects because the movement of water molecules is influenced by both compartments. Finally, anisotropic diffusion refers to diffusion that occurs predominantly in one direction. In the brain's white matter, for example, water diffuses more readily along the length of axons than across them. And this is due to barriers like the myelin and the alignment of the axonal fibers. These properties of diffusion are really critical for understanding how water movement, which is what we measure with diffusion imaging, reflects the underlying tissue organization and integrity, because this all forms the basis for diffusion imaging techniques, including DTI. So it's really crucial that we understand that there are differences in the way that the water moves in different compartments. Okay, so the most common way anisotropic diffusion is conceptualized and quantified is with the diffusion tensor. And this is a mathematical model that assumes water diffusion in each voxel follows a Gaussian or normal distribution. In its most basic form, the diffusion tensor is represented as here in a 3x3 covariance matrix. And that comprises six unique elements, which are these three and then these three. And these six elements describe the magnitude, orientation, and degree of anisotropic diffusion. These elements are derived from 6 gradient directions, which are predefined during image acquisition. And each gradient direction defines the sensitivity of the MRI scanner to diffusion along a specific vector in space. And we call these vectors B vectors sometimes. This information is estimated from 6 diffusion-weighted images, and each is acquired along one of those 6 unique gradient directions. And we also collect one unweighted reference image, and this is called the B0 image. The diffusion tensor can be visualized as an ellipsoid, as in down here, and this is within each voxel. And the ellipsoid provides an intuitive representation of water diffusion. The axes of the ellipsoid correspond to the eigenvectors, which reflect the directions of diffusion. And the major eigenvector aligns with the direction of maximum diffusion, while the medium and minor eigenvectors describe diffusion in directions perpendicular to this. The shape of the ellipsoid is determined by the relative size of the eigenvalues, and these quantify the rate of diffusion along each corresponding eigenvector. So how quickly diffusion is moving in this direction versus this direction, for example, that's going to change the shape of that ellipsoid. So if it's moving in all directions at the same rate, it's going to be isotropic. If it's moving fastest in a primary direction, it's going to be anisotropic. Together, this ellipsoidal representation allows us to identify directional diffusion properties, fiber orientations, and the spatial anisotropy of diffusion. We can derive 4 key metrics from the diffusion tensor using some more or less simple math. Fractional anisotropy, or FA, reflects the extent to which the shape of the ellipsoid deviates from that of a sphere based on the degree to which the 3 eigenvalues differ from one another. So it's like the variance of the diffusion rate throughout the voxel. It changes when there are changes to fiber organization or white matter microstructure, and typically an increase reflects constrained diffusion, and a decrease typically reflects some sort of pathology in the white matter. Mean diffusivity, or MD, is the magnitude of diffusivity averaged across all directions within a voxel. It's sensitive to variations in the ratio of intra to extracellular water concentrations, and those may be caused by cellularity, edema, or necrosis. And an increase in MD may reflect increased tissue water due to edema, while decreases may reflect cell proliferation, for example. Excuse me. Axial diffusivity, or AD, is a measure of directional diffusion parallel to the major principal axis. So in this way, it is sensitive to axonal integrity, such that it will increase when axonal integrity strengthens or neuroplasticity occurs, and it will decrease when axons are damaged or lost. Radial diffusivity is a measure of directional diffusion that is averaged over the 2 transverse axes that are perpendicular to this principal axis. So these 2 right here. And it increases with demyelination, Wallerian degeneration, and inflammation. So in all these cases, the ellipsoid is going to expand. So these are going to get larger, and this one's going to get shorter, and it's going to change as those things change with things like demyelination, for example. Okay, so DTI has had a significant impact in neuroscience within a relatively short period of time, and it is by far the most commonly used diffusion MRI method to date. Its success largely stems from the simplicity of the tensor model and the relatively low requirements on the data needed to fit it. However, it does suffer from a number of limitations that have always been known, but many of which are now recognized to be very serious compounds. One significant drawback of DTI is its reliance on Gaussian diffusion. DTI assumes that water molecules diffuse in a Gaussian manner, which means that they spread out evenly in all directions unless they're constrained. However, this assumption really oversimplifies the complex biological environment of the brain, where diffusion is often non-Gaussian due to barriers such as cell membranes, axons, and myelin. So in other words, diffusion often includes a restricted component. This oversimplification prevents DTI from accurately modeling restricted diffusion, and as a result, it can't isolate or quantify critical microstructural properties like axonal density or fiber organization. This limitation leads to the loss of that critical information of the underlying tissue microstructure. And this is particularly true in areas with crossing fibers or complex configurations, which I'll talk more about in a minute. Another limitation, which is related, but it concerns the sensitivity and specificity of the DTI metrics themselves. So these are like FA and MD. And this has everything to do with the different ways in which diffusion occurs in various biological tissues, like we talked about earlier. So here's that little figure again to remind you. While DTI metrics are valuable for detecting changes in white matter, they are not specific at all to any particular microstructural property. For instance, a decrease in FA could indicate reduced myelination or axonal damage or increased free water content. But DTI by itself is not able to differentiate between these three possibilities. Additionally, because DTI does not quantify restricted diffusion, it doesn't capture that microstructural change fully in areas with significant cellular barriers. The lack of specificity complicates the interpretation of DTI changes, especially when linking them to specific pathological processes or clinical outcomes, which unfortunately is done all the time. DTI metrics are also highly susceptible to free water and partial volume effects, which occur when signals from cerebral spinal fluid or other tissue types contaminate the voxel. Within a single voxel, multiple tissue types may be present, like white matter, gray matter, CSF, vascular tissue, etc. And they all will contribute to the signal. So these contributions are averaged during tensor reconstruction, resulting in a single dominant diffusion direction. For example, voxels in the core of a white matter fiber bundle are less affected by partial volume effects and have a higher FA, while those at the edges of the bundle show reduced FA due to the contribution from surrounding tissues. In this image here, see, these are all only influenced by the white matter, and so they're going to have a high FA, whereas these other ones have some contribution from other tissue types. And that's why when you look at the fiber bundle here, the inside is higher and the outside is lower. And this issue is especially pronounced in smaller fiber bundles, again, as you can kind of see in these images here, where partial volume effects have a greater impact on the average FA compared to larger bundles with more unaffected voxels at the core. All of these issues are further related to probably the most significant limitation of DTI, which is its inability to accurately resolve crossing fibers, which occurs when multiple white matter tracks intersect or run in different directions within a single voxel. The term crossing fiber problem can actually be a little misleading, though, because it really refers to any situation where multiple fiber orientations contribute to the signal measured for a given imaging voxel. So these are just some possible configurations that might be present in a single voxel, although there are an infinite number of different configurations that could be present. The point is that they don't necessarily always cross. They might just touch or fan or bend or something like that. It's really just complex fiber configurations that are unable to be resolved by DTI. Complex configurations are widely present throughout all of the white matter, as you can kind of see down in this image down here. It shows areas with only one fiber orientation. Those voxels are in red. Voxels with two fiber orientations detected are shown in green, and then voxels with three or more are shown in blue. So single fiber regions are essentially restricted to the midline of the corpus callosum and some parts of the cortical spinal tract. And in fact, recent studies have shown that as much as 90% of all white matter voxels contain crossing fibers. DTA has a problem with this because the ellipsoid model only represents the dominant direction of water diffusion. Thus, it only describes a single primary direction per voxel averaged across the voxel. And it assumes that water diffusion within that voxel is mostly uniform. In regions where multiple tracks cross, diverge, converge, whatever, DTI oversimplifies the diffusion profile by averaging them. This leads to inaccurate representations of the underlying fiber architecture and results in underestimations of FA, as the combined diffusion signals from multiple tracks create the appearance of more isotropic diffusion. This is particularly problematic because FA is often used as a proxy for white matter integrity, and misinterpretations can lead to incorrect conclusions about someone's health or pathology of the tissue. So it's really important that it's that this is a issue that we know it has, because otherwise these misinterpretations are made and can have vast implications. The limitations of DTI highlight the need for advanced diffusion imaging techniques. We need techniques that can better account for non-Gaussian diffusion, remove the effect of different tissue compartments, and adequately resolve complex fiber configurations so that we can improve the accuracy and interpretability of results. There have been several advanced techniques that have been developed to address these issues and others. And I will be going over those in the next and last part of this lecture series on diffusion MRI. Thank you, and I'll see you there.